Thanks everyone for joining our webinar hosted by the BetterWater4All network. So this is EPSSC NERC-funded network, and we are one of the 6 EPSSC NERC networks nationwide. Our focus is water, and I'll quickly introduce the network. Around the water issue, we know a lot of water issues in the news in the UK, globally, and our aim is to ensure equitable and resilient access to high quality water for future generations. Just to put that in the context of the pollution, environmental pollution or water pollution, we are facing a lot of pollutant problems. And this group of emerging chemical and biological micropollutants will be the focus of our engineering solutions to ensure future clean water. Through the network, we want to encourage collaboration, co-creation between academia and industry and policymakers and all types of stakeholders, and use systems approaches, not working in silos or looking at a single technology, but not looking at the system. We want to stimulate innovation to have efficient, low-cost, and sustainable technologies for the future of water engineering. And that type of technology needs to meet multiple objectives, not only the efficiency of water purification, but also cost-efficient, social equity, net zero, and sustainability. That being said, we want the technology to be cost efficient, so our water prices from as a water consumer, we don't have like huge water bills at home. And this network is hosted by 4 universities, led by University of Surrey here, and we have University of Newcastle, Cranfield, and Harrier-Watt in Scotland. And we have our 2 lovely network managers, Miriam and Zhu Fang, who are here in the room and who are available online for any queries about our network. So don't forget to subscribe to our network. If you take a picture or scan the QR code, you are able to browse our website and join the membership or our email list for future activities. So our webinar today is by Professor William Gaze from University of Exeter. William has been working in the environmental dimension of AMR for many years, and he has done quite a good amount of research projects, but also influencing policy, the EU and UK and UN policy on environment for AMR. So I'll give the floor to Will.
Okay, great. Thank you very much. Right. I can't see anyone online, so if there are any problems, just unmute and shout out. So I'll assume that you can hear me. But yeah, so thank you very much for the invitation. It's great to be here in Guildford, University of Surrey, my first time. So yeah, I'm going to talk to you about the work we've been doing around AMR from, a One Health perspective, I suppose people like to call it, and particularly focusing on aquatic environments. So first, an apology, anyone who's seen me talk in the last few years probably will have seen some of this before, but hopefully there will be something new, even if you have seen me talk relatively recently. So yeah, I'm based at the University of Exeter, but not in Exeter itself, down on the Cornwall campus, which is near Falmouth. So this is a picture at the top here overlooking the Penryn River and Falmouth Harbour and out to Falmouth Bay. We have a group of about 15 to 20 people working on this subject, quite a wide range of different projects. Most of them have an aquatic element, not all, but a good proportion of them. So I was going to start off with this diagram, which tries to sort of highlight the fact that we have AMR in animals, because of course the majority of antibiotics used globally are used in agriculture. We have the antimicrobial resistance in human populations, but we also have this in the environment around, rivers and streams, drinking water, seas, lakes, aquaculture, whatever. So part of the reason I use this this figure, which is quite often used, and you might have seen before, is because actually, if you look down here, it says after Linton 1977, modified by Irwin, who worked, excuse me, I've got a bit of a frog in my throat, who worked for the Canadian government. And actually, I managed to track down the original figure that this was modified from, which is this figure actually. And so I sort of wanted to show this because those of us that are sort of working in this area, we all think that One Health is quite a new thing, and people think, well, we've got to really focus on these intersections between humans, animals, and environment. And actually, I like this figure here from 1977 better than the new ones because it's probably arguably simpler. Also, because actually these arrows that it has here, these black arrows and the white arrows, are indicating antibiotic selective pressure. So this is the action of antibiotics on bacteria that drives the increases in resistance. So I've made one modification to this, that I've put this white arrow here, which is, and white is low and black is high in this figure. So I'm suggesting that the only difference really that we now know 50 years after this figure was produced, it wasn't called One Health 50 years ago, is that we now know that there's a possibility or probability that some antibiotics in some circumstances might also be exerting a selective pressure in the aquatic environment itself. But otherwise, this is pretty much similar to everyone's talking about now. So it's good that that's a consistent idea of messaging. But on the other hand, it's a little bit disappointing or discouraging that 50 years later, we're still talking about the same thing. We're still saying we need to understand better the link between AMR in animals and AMR in humans, or AMR in the environment. What effects is it having on, what's this dotted line going from the sea to man with bathing, on it? What is that doing in terms of the burden of resistant infections? The answer is we don't really know. The full answer, we know more, of course, than we did in 1977. But again, this brings up questions about how do you act in the absence of certain evidence, on something that's very pressing, like antimicrobial resistance. The other slide that I always show is this slide, which looks a bit incomprehensible. But this is the origin of Earth 4.6 billion years ago. This is the timeline of life on Earth to the present in the top right, and then if you can see in some of these labels here, this one says origin of B3 metallobeta-lactamases. So that's a group of enzymes that breaks down antibiotics, and then it's got things like origins of streptomycin here, or origin of something to do with probably cell wall structure or something like that. But anyway, what this is really telling us is, to the best of our knowledge, we know that antibiotic resistance has been around for 2 billion years or more. So it's a natural process, but one which, of course, the evolution of resistance has been massively sped up and amplified by the amount of antibiotics that humans have produced, most of which, of course, are natural products produced by microorganisms. But when I think of antibiotic resistance, I think of it in two different ways. So there's this long-term emergence of antimicrobial resistance. So this is a little bit analogous to emergence of zoonotic infection, like SARS-CoV-2, which causes COVID. That's a virus which emerges from a wildlife reservoir, but resistance can emerge from a natural environmental microbiological reservoir, because you might not all be aware that resistance genes can move between even unrelated bacteria. So resistance doesn't just evolve within the clinic. We know for sure, for certain, that the origins of much resistance that's currently in human pathogens, and animal pathogens for that matter, are of environmental origin. But what most people are thinking about when they're thinking about aquatic systems I think it's this acute transmission. So how many people get ill from exposure to aquatic environments, and how many of those infections are caused by antibiotic resistant bacteria? So those are two related but quite different things, I think. And when we're thinking about certain elements of water treatment or the water cycle, or agricultural pollution that goes into river systems, potentially they could be driving both of these, or one or the other, or neither. So, and again, for those of you that are familiar with this sort of ideas around One Health, which the archetypal one is these three circles that sort of overlap with the, in the center you've got One Health, which is environment, human and animals. This is a little paper that I was a co-author on, where we suggest that an alternative way of thinking about One Health is not that the environment is something separate that overlaps a little bit with animal and human health, but in reality, because of this reality that most of the resistance, the transferable acquired resistance we see in bacterial pathogens now, it comes from the environment. We should really think about, and of course this is literally, we are encompassed by the environment. So, the environment is central to the AMR story, not just something added onto the end. So I'm going to talk really in these three different areas today. One on sort of these landscape scale processes and how aquatic systems are involved in dissemination of resistant organisms, much of which, of course, comes from humans and animals. But of course, we have to remember there's this environmental resistance there as well. And then there's an environmental exposure and transmission. So the risk to human health from that environmental reservoir, whether that be the natural environmental reservoir or the anthropogenic AMR signal produced mostly by the way that we deal with our waste. But then over and above that, there's this capacity or potential that antibiotic residues, that when we take an antibiotic or when an animal is given an antibiotic, or in the case of antifungals, which of course are sprayed onto onto crops and things in the environment at effect concentrations, treatment concentrations. Also in aquaculture, we might see antibiotics used as treatment concentrations in the environment itself. But whether all of that is producing antibiotics at sufficient concentrations to drive this enrichment or generation or selection for resistance in the environment itself. So first of all, people have been talking about this obviously for a long time. I showed you that slide, or that figure from 1977, which is showing the fact that this is the system where resistance is circulated. But probably I've been working in this area for about 25 years, and this paper on the left, this was UK Health Security Agency or Public Health England, whatever it was called, Health Protection Agency at the time. And they took a couple of water samples from the Thames and found E. coli that produced these beta-lactamase CTXM14, which was a really important group of resistance mechanisms. And so they concluded that this could have been transient. They didn't really know whether these were resident in the aquatic environment, but it was notable because these CTXM genes, as they're called, these have a clinical signature. We don't find them in environmental bacteria. They are mutations of a naturally occurring gene which was mobilized into clinical pathogens at some point in the past. But at about the same time as that, we were also looking in rivers. So this is about 2009, something like that. And we started looking in sediment rather than the water. So we took sediment samples upstream and downstream of a wastewater treatment plant, and then used a variety of different methods to look at what the resistance profiles of those communities of bacteria were in that sediment. And what we found was there were lots of clinically important resistance mechanisms in the downstream sediment. There were some, of course, also in the upstream sediment, but the effect of the plant, so the upstream-downstream comparator, was very marked. So, for example, again, these are old figures because it's from quite an old paper. But what this shows is in black is downstream and white is upstream. And on the left here, this is E. coli that are resistant to this particular type of important antibiotic, so third generation cephalosporins. And in this case, so yeah, this was the number of resistant bacteria. So downstream, upstream in 2011, and then these are non-E. coli coliforms, so other coliforms, and then downstream, upstream. So you can see that the number of these resistant organisms was really quite a lot higher downstream. And then this is actually, these are sort of cartoons of the bits of DNA that surround the particular type of resistance gene. So this is one of those CTXM genes. So in this case, CTXM15, and these are different genetic contexts. So the CTXM15 gene are these spotty ones here. Sometimes they're duplicated in quite a few cases, but something like the majority of these genetic contexts were novel, so they hadn't been seen in any clinical strains. So we thought that this could be evidence of selective pressure, that we are selecting for all of these different insertions, because these genes are actually mobile. They're mobilized by this insertion sequence, which is just upstream. So they can hop in and out of DNA. So this is evidence that there are lots of different genome contexts that are being selected for, because these genes usually confer a fitness cost if there's no selection, no antibiotic present. And then we also use a different method. So we use quantitative PCR to look at a marker for resistance. So this is percentage. So 2% doesn't sound much, but then that's probably 2% of a billion bacteria per gram. So 1% of a billion is 10 million. So that would be 25 million bacteria per gram carrying a copy of whatever these genes are. And this is the integrase, an integrase gene that's associated with a genetic element that can integrate many different types of resistance genes to all different types of antibiotics. And then down at the bottom, this is downstream one. So this was probably 100, 200 meters downstream of the point where the effluent discharged into the river. And then I think this is about 500 and then 1,000. And this is upstream. So this is a molecular marker for resistance. And we can see there's a marked effect there as well. And then with about this time, I met Andrew Singer at the Centre for Ecology and Hydrology. And we did this across the Thames catchment, where these red dots are where we took samples. This is the prevalence of that marker for resistance at these different places. So Thames at Wheatley, Leach at Lechdale, Holme at Whetford, so there's all these different sites in the Thames. And you can see that the range of these of the signal is quite different in the different sampling points. And then we did so that's 4 samples over, I think, 2 years. We're trying to be sort of seasonal. And then what we did was we looked at, so this blue triangle is the sample site, and then these are the treatment plants, I think, in a 10 or 20 kilometre radius upstream. The size of the plant marker is the population served, and then the colour is the type of the plant. So then we did some simple statistical modelling, and we could predict about, I think, 80% of the variance in this marker between the actual and predicted. And about 50% of that variance was associated with point source pollution, so wastewater treatment plants, and 30% with diffuse. So that was more associated with the type of land use. But the things that really were driving this were the distance from, size of, and type of plant. So we can basically see across the Thames catchment that the levels of antibiotic resistance in those sediments is strongly associated with wastewater treatment plant location, size, and level of treatment. So I'm going to move on then to the next bit, which is about, well, okay, so we know there are resistant organisms in aquatic systems, but what threat do they pose to health? So this is onto that sort of the so what question, which is what we often get asked, and this is the most difficult question probably of any to answer for various reasons. So we can do exposure risk assessment. You know, maybe really what we want to know is like how many people get ill. But most of these pathogens we're talking about are opportunistic pathogens. So they not unlike an obligate pathogen like a virus, or most viruses, let's say, COVID, of course, could cause asymptomatic carriage, but people are actually actively infected. The thing about these E. coli is that maybe, yes, you ingest it, it goes into your gut. It doesn't do any harm to you unless it gets somewhere like into your bladder or into a wound or into your bloodstream, or potentially into the bloodstream of a young child or an elderly parent that we might be in contact with. So this sort of classical outbreak tracking for opportunistic bacterial pathogens is very, very difficult. But you can do an exposure risk assessment where you look at the levels of resistance, you then estimate the volumes of water that people might ingest, and then you can sort of, you can work it out that way. So my colleague Anne Leonard did this we took Environment Agency samples for enumerating E. coli, and then basically we estimated that there was about 6 million recreational sessions in 2012 in England and Wales where with ingestion of one or more of these third-generation cephalosporin-resistant E. coli. And actually, if you extrapolate that to all resistance mechanisms, that number goes up hugely. Basically, every time you go swimming, you will be ingesting a resistant E. coli. Again, what that means, we're not so sure. But one way we thought we could try and find out what that meant was, so we started looking in, we thought we'd look in the gut microbiome of surfers. So this is this beach bum survey, we called it, which has had quite a lot of traction. This was a box that was in the National Action Plan, where I think they said there was 1,000 research papers published on AMR in the water environment in the previous 5 years. This was the only one that was cited, maybe because it's the only one trying to answer that question, what are the health outcomes? So this basically, we found that surfers were 3 times as likely to carry these particular types of antibiotic-resistant E. coli in their gut microbiomes. That's still not how many people are getting ill, but of course we know that colonisation is a risk factor for infection. We're now doing that work in freshwater swimmers in rivers, which of course, there's concern about rivers, as many of you will be more aware than I am, because of course that there's less dilution in rivers than in the sea, so there's potentially more higher levels of resistant organisms that people will be exposed to. So Elitza is currently, she's done that study, she's on maternity leave, and she's analysing and then writing the paper up when she gets back. So in terms of illness, the evidence arises around things like, big organised water sports events, where we see people that do get ill, people that have had near drowning events and then get, respiratory infections with AMR bacteria. But this paper actually looked at risk factors for UTI and found that recreational freshwater swimming had an odds ratio of 2.1. So you're twice as likely to suffer from a resistant UTI if you're a swimmer than if you weren't. That doesn't sound much when you compare it to an odds ratio of 21 if you've been abroad to Southeast Asia or Africa in the past 6 weeks. But then if you look at the odds ratio, if you've been abroad in the last 6 weeks to 24 months, that's 2.3. So that's actually almost the same. So then you start thinking, well, maybe actually it's not such an insignificant contributor when you think about all the different drivers for potentially causing infection. And there's another paper here where they tried to do some sort of attributing sources of community-acquired carriage of particular resistant E. coli. And I'll just, if you want to read this, then it might be worth reading. But what they did was then they tried to attribute the percentage of cases that were to do with human-to-human transmission, so 60%, seafood consumption 6%, and then we've got dog, chicken meat consumption, swimming down here at 2%, and then turkey raw consumption, being a farmer, all these things. So what Anne then did is for the summer months when people are more likely to be swimming, she extracted the data and reordered it just for the summer. And at that point, you start to see, well, swimming comes out at 6%. And if you combine that with seafood consumption, which may be also because of aquatic pollution that are accumulating within, shellfish or whatever, then that's 13%. So that becomes quite a, that's still only 13%. But if that's 30% of all infections, which are killing thousands of people a year potentially, even in the UK, and millions of people globally, that is something to consider. But that's really where we are with that. We are thinking about how would you then do a trial to work out numbers of infections. But again, because of the opportunistic nature of most of these pathogens, it is really challenging. And this is a review paper here where we start to discuss some of these complexities. But I won't go into that because I will run out of time. So that's the first bit of the talk. And then I'm going to go on to this the final bit, which is, so what about this issue of in situ selection? We know that what we do as a society enriches resistance in the environment. We know that people are exposed to those aquatic reservoirs of resistance. We know that they include critically important human pathogens to, high priority antibiotics. So we know they're exposed, and we know people that can result in increased colonization. Not every study shows that, but some do. And also there is this evidence around associations with increased risk of infection. But then what about whether the antibiotics that are also present in the environment are driving resistance in aquatic environments? So this is where we get on to talking about predicted no effect concentrations of antibiotics, or minimal selective concentrations of antibiotics. So this is the Water Framework Directive hazardous compound watch list that used to be based on sort of ecotox, acute toxicity, but has begun to be informed by the potential for these antibiotics to select for resistance in the environment. So very quickly, there are different ways of estimating that. Some can be based on the minimum inhibitory concentrations of those bacteria, of human pathogens to antibiotics. Some are based on experimental data where you might take a resistant strain and a susceptible strain, grow them in the presence of an antibiotic and see which one does best, and you can work out what the minimal selective concentration might be from that, or using complex microbial communities studies, which is what we do, which has this has the reservoir of resistance mechanisms that can move between different bacteria. So again, I might not go through all these slides in detail, but it's being recorded, so you can go back and look at them in more detail. But essentially, again, we think of this being quite a new thing, but in 2003, which is now scarily 23 years ago, Klaus Kummerer was predicting that, in fact, yes, if you looked at the measured environmental concentrations of antibiotics in wastewater, and you used an MIC 50, so the concentration of antibiotic where half of susceptible pathogen bacteria might be inhibited, the ratio between the predicted environmental concentration and the predicted no effect concentration for hospital effluent could be 10 or 20 times higher than the MIC 50. So he concluded that means that perhaps the hospital and even municipal sewage could be a selection pressure, could exert selection pressure on bacteria. And then in 2011, there's this Swedish group that started doing this experimentally, where they had a susceptible and a resistant strain marked with different fluorescent markers, and they grew them in the presence of different concentrations of antibiotics, and then they could work out what the lowest concentration that favoured the resistant one was. So not killing them, but altering the growth rate, which means that over time, the resistant strain would outgrow the susceptible one. And then Johan Bengtsson-Palm and Joachim Larsson, and this is the data that now informs things like the Pharmaceutical Industry Alliance and potentially the WHO thinking on this, is using the lowest 5 percentile of MICs of all susceptible pathogens. But those pathogens, of course, are grown at 37 degrees on an agar plate in a lab, and they're human pathogens, not environmental bacteria. So you can see already there are these different ways of trying to determine these concentrations because it's very, very, very challenging to do it in the environment itself. So essentially, this is what I was showing you, that in red, we have increasing concentration of antibiotic going from left to right. And as the concentration increases, it inhibits the growth of the susceptible strain to the point where it kills them. And that's the MIC susceptible. And then the resistant strain survives longer at the higher concentrations and then outcompetes. But where those two lines cross, that is defined as the minimal selective concentration as opposed to the minimum inhibitory concentration. And then this is getting into the weeds basically a little bit, but essentially what you do is you establish growth rates at different concentrations. You then use those, so you use this growth rate to produce a selection coefficient. And where you plot those selection coefficients, where it crosses the x-axis, that is defined as the minimal selective concentration, which corresponds to this point here, where the growth rate of the susceptible and resistant are the same. And in this paper, they were defining that concentration for ciprofloxacin down to nanogram per mil, which is picogram. No, hang on, that's the wrong way around, is it? I think it's so nanogram per mil. Yeah, so it would be microgram per litre and then nanogram per mil. I think perhaps it might have been even lower than that in one instance, but I'm not seeing that here. But basically, for those of you that know what concentration of antibiotics are found in the environment, they can be high nanogram per mil into the high nanogram per litre to low microgram per litre. So if you're having the MSC right down low in this range, it's confusing because of the units. That means there's the potential that antibiotic at that concentration would be selecting for resistance. So we then thought, well, we'll do this in the complex community, use like sequencing and qPCR and things like that to look at the fate of resistance in complex communities exposed to different concentrations. So this is the sort of what the output we generate. So this is like this is a whole concentration range of ciprofloxacin in microgram per litre, and then we see that the lowest observed effect concentration is 15. So that is still quite high relative to what we might find in the environment. That's probably lines up with the sort of the highest levels that are measured in the environment. But remember, this is for a single compound. If we're thinking about antibiotics in the environment, they will be often in complex mixtures. Sometimes multiple antibiotics in the same class, and then you have like the effect across classes. So we're also doing work on mixtures. Is that additive? Is it synergistic? Is it antagonistic? So then you sort of you end up, and this is an example for ciprofloxacin. So in our complex communities, we see this lowest observable effect concentration is 15. So to get a PNEC, you then look at the next concentration down, which is the no observable effect concentration, that's 7.8, and then you apply a safety factor of 10. So your PNET, your predicted no effect concentration comes down to 0.78, and then that microgram per litre. So then that is well within the range of the sort of these higher concentrations of this single drug that we see in aquatic environments. And you could do that for the MSC, you get a slightly different outcome. And then the ecotoxicological PNEC, which is like the toxic effects on blue-green algae, is 0.089. So probably that's like one order of magnitude lower than the complex community PNEC. And then you see the same for the MIC-based PNEC, it's like 0.06. So there is some difference between those, but within an order of magnitude, I suppose. So we can argue which are most protective. Well, this is the most protective, but which is the most realistic. But whatever the argument is, those concentrations are pretty low, and they are in the range of concentrations that we find in aquatic environments, and certainly within wastewater treatment, and also within animal manures and even probably soils that have had animal manures applied. So then we end up in the situation, it gets a bit complicated, but we think, well, this is indicating selection for resistance. So we have this positive selection for resistance where we have concentrations of antibiotics above this minimal selective concentration. This is the minimal selective concentration where the resistance persists over time. But then actually we noticed that even below that level, you can have this decreased levels of, or you can have these decreased rates of decay, even in concentrations that are below this minimal selective concentration. So compared to when there's no antibiotics itself. So this is really telling us that even really quite low concentrations of antibiotics, even below the minimal selective concentrations, might be impacting the levels of resistance in the environment and potentially emergence of new resistance. This, of course, is not completely verified in the environment itself, but again, it's quite challenging, you know, without using genetically modified organisms or whatever. But people are trying to do that. And you can, there are summaries of this type of work. This was a paper that we did as part of the PathSafe project, which was the government-funded 20-odd million pounds AMR project. So this is published with Environment Agency staff. So if you're interested in the subject, do go and look at that paper. This is just a quick, I'm coming towards the end now. This is a quick example of where we've applied actually the data from the SIP2 and water industry program, looking at antibiotic concentrations in wastewater and applying this sort of environmental risk assessment for ciprofloxacin. So this is based, so the colour is what you think. Red is high probability of selection, orange medium, you know, green low. So this is in influent, this is in effluent, and this is in surface water. This is for our experimentally derived data. If we use the Bankston Palm MIC derived, of course, this risk would appear higher. So again, you know, but it's just, this is how we go about the risk assessments. This is just looking at across different parts of the country. You know, we see that it's somewhat different for the different regions. So in summary, these MIC-based PNECs that are derived from susceptible pathogen MICs are extremely useful for informing risk assessment in aquatic environments, but we need to use these experimental methods to double check those, and also we can ask other questions. You know, so when we're doing our community experiments, we can play with temperature, you know, whether climate change might change things, or we can change nutrient levels. You know, we can actually try to model different scenarios which might be driving resistance. There are these reports from the United Nations. This last one on the right, Racing for Superbugs, Strengthening Environmental Action. That's a really good overview of the whole subject. If you want to go and look at that, I put them on the left because I co-authored that one. That's much simpler. It's a little bit out of date, but it's still pretty current. And then at the moment, this is a bit of self-advertising that we have a network as well. Not similar size, I think, maybe to Water4All, about 250 people. And this is really, there was 8 different networks funded on antimicrobial resistance the year before last by UK Research and Innovation. And this one is on looking at the impacts of climate change, but also environmental pollution. So this is the one that incorporates the most of the environmental sort of dimension of resistance of those 8 networks. And that's people in the group that contributed to this work, of course, because apart from the very early stuff when I was a bit younger, none of it I've actually done myself. Okay, thanks very much.
Have you seen any questions coming up on chat?
Yeah. Oh, so yeah, there's hands coming up.
There's either the Q&A or the chat, but I haven't seen any.
How do you unmute? Someone raised the hand. Anna.
So can we unmute everyone?
Or you can unmute your own.
So people can unmute themselves.
Yeah. So while we solve the technical online things, probably let's see if there's any question from the room.
I can ask a question.
Yeah.
Will, can I ask a question?
Yeah.
Yeah. So I was just wondering what your reflections really are, right? Because you've described a situation where you mentioned cross resistance, right, where there's multiple drivers, non-antibiotic based drivers of selection. So I wonder how we deal with that complexity really in thinking about AMR. Will we ever get to a situation where we can stop selection of AMR?
No, I don't think so. I mean, as you know, we know it's a natural process, isn't it? You know, there's so many things that can indirectly select, because you know these genetic elements, these plasmids, they can have biocide resistance genes on, metal resistance genes on. We now find out that something like 25% of all pharmaceuticals have some antimicrobial activity. There's evidence that antifungals might even select for antibiotic resistance, you know, albeit at low level. Pesticides, herbicides, you know, there's some evidence for that as well. So I think we fixed the unmuting issue.
But people have to unmute
So
themselves.
but
Not everybody can talk at the same time.
no. So it's about reducing the rate at which resistance develops, so we can keep one step ahead in terms of drug discovery, which we know, of course, that there's less new drugs being brought to market, partly because resistance wipes out your $1 billion investment, you know, overnight, yeah, in a few years. So yeah, I did see some hands go up. I can't see any hands, but I think there were some up.
No, I can't see.
People
any
still
hands
not
up.
able to
Otherwise-
unmute.
You know, people are, but
Yeah,
people have to
I
do it
know.
themselves because you don't want 80 people all unmuted at the same time.
Okay.
So if anyone online has any question,
Yeah, just unmute and
yeah,
ask your
unmute.
question.
and if you want to type in the chat box, that's in Q&A box, that's also a channel.
I think you were so clear and well presenting that nobody's got any
Crystal
additional.
Ruth is
there,
asking a
yeah.
question.
Hi, good afternoon. Thank you for the presentation. I was thinking the presentation is focused on wastewater treatment. I imagine partially we have so much data, but I have this sunk feeling, this feeling of what are we going to do, because since privatization, a lot of the water has been treated. The waters are cleaner than they were in the 80s, and now you discharge into coastal waters, you have UV to try to maybe not kill the bacteria, but the bacteria will stop being able to reproduce. However, just because you treat with UV, that doesn't mean that the DNA, the RNA is not able to carry on being part of this antimicrobial resistance problem. So what do you envisage as the next steps in water treatment? It's just that I think, okay, we know bacteria come into the treatment works, obviously come from our excrement and things like that. Of course it has bacteria. But even when we treat it and we put UV, that doesn't mean that that bacteria cannot carry on doing its thing.
Yeah, no, you're quite right. You know that in theory, DNA from dead cells can persist and can be taken up by other cells. But I think, you know, the risks associated with those have got to be significantly less than those posed by live organisms. So I suppose that's one thing. You know, I mean, the way we have to address this is we also need to reduce the usage of these things, so there's less going into treatment plants. Most treatment plants, of course, don't have disinfection, so you know, maybe we're reducing numbers of E. coli by 1 log or 2 logs. But you know, I think I couldn't find the study which worked out the actual load of resistant organisms going into UK aquatic systems every day, but it's a lot of zeros. It's like 10 to the 20 something, you know, a day. So that's the thing about microbiology, I suppose, you know, in the microbial world is that, you know, we can say we remove 99%, but you know, if you remove 99% of 100 million, you've still got 1 million left, haven't you? So
yeah, absolutely.
the
Yes.
numbers are really high, yeah. Well, I think there are technologies, aren't there, that are available, which have been, and you know, but they're expensive and they consume a lot of energy. So you know, we're still, I guess we're still at that point where we're trying to work out still, hopefully in 50 years' time, we're not still having the same debate. Like, 50 years ago, we were having the same debate about animal work. But, I think, and we know that, of course, there are raw sewage discharges. There always have been, and there are at the moment too. Some of the work we're doing is looking at the differential fate of resistant pathogens from treated and untreated wastewater, because actually you see different diversity of E. coli in treated rather than untreated, because of course the treatment is
Yeah, of course.
environment of itself. So maybe you're selecting for particular types, and it may well be, and some evidence suggests that you maybe we're selecting for things that are less resistant, because if resistance confers a fitness cost, if there isn't selection within the wastewater treatment system, you might hypothesize that they should be less resistant. And I think that where it gets complicated is that you see that actually for many resistant mechanisms and those bacteria they're associated with, they do decrease in relative abundance between absolute abundance. But there's always some that do the other, go the other way around. So I think that we know that probably selection itself is not responsible for the bulk of those bacteria that are in aquatic environments. They're coming directly from waste. Of course, without wastewater treatment, they would be far more numerous. So we always have to acknowledge that, if we're removing 90 to 99%, that is a lot better than, of course, nothing. But of course, it could be we could be doing more. In Sweden, they're using membrane filtration, aren't they, on new plants? But again, they have maybe lots of money and
they are 8 million people, and we are nearly 80, maybe. And
that's right. Yeah.
Yeah.
So it's about as a society whether we're willing to pay that cost. That's right.
Yeah. Thank you.
So I'll read a question from the Q&A. Other than novel antibiotics, what do you think are the most valuable interventions?
Yeah, that's a good question. For treating resistant infection.
Overall,
I
think.
I mean, there's lots of interest, aren't there, in things like probiotics and prebiotics. So, that prebiotics, the idea that what you eat alters your gut microbiome, and, your gut microbiome, the diversity in the health of your gut microbiome affects the probability that a bacterium in your food or water, whatever, will actually be able to colonize your gut. So, there's evidence, of course, that so the higher the diversity, maybe the less likely they are to be invaded by foreign bacteria. Then probiotics, of course, is actually consuming live bacteria that might then block the colonization. Of course, in terms of treatment, there's still lots of interest in phage therapy. There's interest in all of these bacterial immune systems that we're finding that exist, like CRISPR systems. So CRISPR, which now we use to genetically modify human DNA, is originally developed as a natural immune system in bacteria, which recognizes phage, or viruses that infect bacteria, and can chop up the phage and so protect them from infection themselves. So people have been using that system to target pathogens or target resistance and actually try and clear it from microbial communities. So we might imagine in the future, if we can get over the sort of the safety and ethical things about consuming genetically modified DNA mobile elements, or even or bacteria that carry these things that can then pass to other bacteria, we might be able to remove resistance from microbial communities and pathogens. So I think there's a whole array of different approaches looking into this, but again, at the moment it still seems to be that we want more drugs, even though of course the numbers of new antibiotics coming to market are relatively low. But yeah, there's a lot of interest in alternatives. More vaccines, but then that doesn't seem to go down well, certain parts of the population.
So there's also a question in the chat box, and Ed helped to answer the question. Hello, Ed. You're online there.
Hi, Ed. So it's asking how significant is waterborne acquisition of AMR compared with hospital foodborne acquisition? Yeah, I think that's a good point. I suppose it depends where you live in the world, isn't it? So if you live in a part of the world where drinking water is not as clean as our drinking water, then maybe it's more important. So these high-level association studies between all different variables, Ed knows this, he's just making me say it, all of these different socio-economic drivers of AMR. So I think it's that transmission, and much of that is attributed to lack of investment in wastewater infrastructure, is driven by potentially by waterborne acquisition. So in low middle income countries, some low middle income settings, maybe it's the majority of transmission. We have thought about trying to do these back of the envelope sort of estimates of relative probability of transmission. The interesting thing with hospitals, it's a relatively small population in hospital. So even if the probability of transmission is higher for any one individual, at a population level, maybe it's not so significant. But of course, those strains that people carry in hospitals are often the really ones we're concerned about. So again, I'm sure that foodborne acquisition of AMR is massive and hugely underestimated, and a lot of the argument that's being discussed, as again to try and confound these links and reduce, impacts on antibiotic usage in animals. We say, well, it's different strains of salmonella that cause infection in humans and in animals, whilst we completely know that the resistance can move between different strains very, very readily. So I think it's not biologically credible to think that the majority of antibiotics that are used in agriculture, and that it does not have an effect on the emergence of resistance in human pathogens. And the more there are some excellent case studies where we see, so livestock acquired MRSA, colistin resistance from the Chinese use of colistin. We know that it happens. We know there are something like between 100,000 and 1 million cases of foodborne food poisoning a year in the UK. So how many people are ingesting harmless commensal bacteria, all these opportunists that don't cause infection, but that have transferable resistance? We're all consuming those things probably daily, I would say. So huge. So yeah, it depends on you, doesn't it? Where you live, what you do, what the relative risk will be. But all we're saying is we're not saying it's the major risk. It could, of course, be quite important in this emergent story, because in natural environments where you've got lots of indigenous environmental bacteria mixing with human and animal associated bacteria in the presence of this complex mixtures of antimicrobials and chemicals with antimicrobial properties, is that where these important transfer events occur, or do they all occur in the human gut? That again is something that we are trying to look at in more detail. And when I say we, I mean the whole scientific community.
There's another one in Q&A.
Interventions.
No, the one about is focused on the exposure risk to humans, which is understandable, but should the risk to animals also be considered? Yes.
Yeah, definitely. I mean, I think so if you think about where water comes for livestock, I mean, we still have, people say it doesn't happen, but of course we have livestock that are drinking from open water sources. That still happens, I suppose. The argument about wildlife is an interesting one. If a wild animal gets ill because it's been exposed to a pathogen, that's one thing. But if it's exposed to a resistant pathogen, no one's ever going to treat it with antibiotics. So does it have any effect? I don't know, probably not unless the resistance is somehow associated with virulence, which potentially it could be in some instances. But no, you're quite right. And of course, companion animals, that they are essentially members of human families, and whatever your dog is colonized with, the chances are that you will also be colonized with it, which is why feeding raw food to for dogs, for example, is associated with higher levels of carriage of resistant organisms in their owners. So again, I wouldn't feed my dog raw food for that reason. Maybe if it was pasteurized, you might consider that. But yes, we do tend to, I suppose, always emphasize the human side, because that's what gets action, and that's what focuses politicians' minds. But we should be thinking, let's see more. Is there more there? Oh, and if there is low exposure risk, does that mean these high levels of resistance are acceptable in the environment? Well, I guess, yeah, that again is a good point. But maybe not many people were exposed to those, those bats or civets or whatever, where, if you think about a global pandemic of any sort, the actual number of those initial transfer events that mean that something that was not associated with human populations is now associated with human populations. They may be relatively infrequent, but they can be incredibly important. So, how many transfer events of COVID have there been? I mean, there have been tens or hundreds of billions, maybe, of transfer events. Maybe a handful of those, or a very small percentage of those, were the original environment or wild animal human transfer events. And I think the same can be said, and that's why it's difficult. So maybe the number of people that get acute infections from environmental exposure may be relatively low, but what effect is that exposure having on resistance within the meta human microbiome? So our human microbiome as a population. Because then it gets spread between people, doesn't it? So I think you've got to think of it in that sense as well. It's not just you go swimming, do you get ill or not? It's about how resistance moves from environmental microbiome, the environmental microbiome, to the human microbiome. Some of those events may be rare, but they could be hugely impactful. So where did all these mobile carbapenem resistance mechanisms come from, or where did those, maybe they emerged in one person in one place in the world, and they formed their own pandemic.
There's a comment from Ed in the chat box, and that would be our last session. Yeah.
Yeah, so I think this is something that the European Union are trying to do now, isn't it? I think I wasn't involved in it, but there's a trial of using qPCR to look for prevalence. I suppose what you could do is you can think about, what are the risks associated with exposure? So, it's complicated because you can't do quantitative microbial risk assessment because we don't know what the dose response is for most of these. So how many resistant E. coli do you need to ingest for that resistant E. coli or the resistance genes it carry to become embedded within your own microbiome, and then of course be a risk factor for infection for you or your people that you come in contact with? So we don't really know that, but we might say, well, we might put an arbitrary threshold, like I think we were discussing this today, actually. So we know what the infectious dose is for some things. It can be, sugar toxin producing E. coli, it's like 50, but for salmonella it's 100,000. So maybe you could come up with, well, this is a range of numbers, or this is some sort of threshold, and everything that exceeds that threshold, you say, well, that's unsafe and it's not safe. Of course, the answer is, of course, we don't really know, but the lower the better. But of course, we can't get rid of everything. So yeah, that's a debate. But I suppose that debate is being had in the Joint Research Centre in the EU after they've had the data back from their pilot study of AMR prevalence in surface waters in Europe. And I don't know what the answer is that they come up with. And I don't know what the quadripartite committee on integrated surveillance are thinking about those things either. I think it's difficult. That's why we never hear what they've decided, because they can't decide.
Okay. All right. Yeah. Thank you everyone online, and thank you, Will, for the nice presentation discussion. We'll finish our webinar.
Do you want to put the last we share again? The last slide on.
Okay. Yeah. So for people who are still there, and am I sharing?
No.
No, you are sharing.
Well, we're over
But
time.
you've got the
Yeah.
wrong.
No, not.
Yeah.
Yeah, that's the one.